Hello, welcome to the Robert Jenkins Microscopy Facility at the University of Wyoming. Its mission is to assist researchers in their microscopic imaging needs and to enhance students' learning experiences. It is located in the Biological Sciences building. The facility has both optical and electron microscopes, which can reveal from macro to nanoscale structures. Let's start the tour with the optical microscopes.
Here you are in the room of the Zeiss zoom microscope. This is essentially a high-resolution stereo dissecting microscope. It has a large field of view and 16x optical zoom that allows you to see structures at cellular level. It has motorized X, Y, and Z stages and is equipped with both bright field and fluorescence. It also has an optome, which is a specialized structured illumination device that allows you to make optical sections on this microscope.
We are now in the room of the laser scanning confocal microscope. This facility has two. The one you are seeing here is a Zeiss 700 model. It is an upright microscope that works well with fixed samples. It has 4 lasers ranging from 405 nanometers to 633 nanometers. The way a confocal microscope works is that a small aperture is placed in front of the detector. The aperture is confocal to the focal plane of the sample. This way, the out-of-focus light will be blocked and therefore making the image sharp. It also gives the ability for a so-called optical sectioning. You can image the same sample at different focal levels and then build a 3-dimensional structure. You can also tile multiple images together to build a high-resolution image from a larger sample, for example, the entire mouse brain.
Here is our second laser scanning confocal microscope, the Zeiss 980. It's a much newer model than the 700, and it's an inverted microscope that is capable of imaging live cells. It contains 5 different laser lines, 3 fluorescence detectors, and 1 transmitted light detector. This microscope is equipped with a super-resolution module called ARRIScan 2. It uses an array of detectors that offer higher speed, higher sensitivity, or higher resolution compared to a regular confocal microscope.
The confocal microscope is great in generating high-resolution images. However, if you need to image a lot of similar slides, it becomes very time-consuming. The slide scanner, on the other hand, is specifically designed for high-throughput imaging. The slide scanner can load up to 100 slides and scan them automatically. The reason it is called a scanner is because it will automatically recognize samples on each slide and tile scans the samples. Then it will stitch them together to form a high-resolution single image.
Now we are in the 2-photon microscope room. 2-photon microscopy uses 2 lower energy photons instead of 1 high energy photon to hit the same fluorophore simultaneously. This is like having 2 people instead of 1 pushing a vehicle. Each person will only need half of the energy. The advantage of the 2-photon microscopy is that the lower energy photons cause less damages to the cell and penetrate deeper into the tissue. Our 2-photon microscope is specifically designed for in vivo imaging of live animals. You can see that while the animal is completely awake and active, we can image its neural activities and correlate the neural activity with, for example, the movement of the pupil.
Let's move on to electron microscopes. As the name indicates, electron microscopes use electrons instead of light as the illumination source. This can produce a much higher resolution than light microscopes. There are 2 types of electron microscopes, a scanning electron microscope, or SEM, and a transmission electron microscope, or TEM. In our facility, we have a Hitachi tabletop SEM. It provides limited resolution but requires little sample preparation. For example, you can simply pick a pine needle, place it on the sample holder, and image it directly. The SEM can be used for viewing surface structures of various samples, such as insects or plants.
While SEM observes the surface structure, TEM examines the internal structure of, such as a cell. The Hitachi H7650 TEM we have in the facility can reach an ultimate resolution of 0.2 nanometers, and the magnification can be as high as 600,000 times. Imagine if I am being magnified for only 300,000 times. When I set my feet in Laramie and lie down, my body will cover the entire Wyoming, and I can use Grand Teton as my pillow. With such a high resolution and magnification, we can easily see nanoparticles, viruses, or subcellular structure like mitochondria.
This concludes the major instrument we have in the microscopy facility. Thank you for coming along with us today. If you need any additional information about the facility, you can visit our website at www.uwyo.edu/microscopy or email us at microscopy@uwyo.edu. Thank you.